what I'm going to talk about today is an evolving piece of work that has really been more than 20 years in the making. Actually, it's closer to almost 35 years in the making. But it's kind of centered on this. These are biopsies of a patient with head and neck who's going to be on a clinical trial, or who is on a clinical trial that I'm going to talk about as we get to the end. But essentially, you can see all the ugly-looking cancer cells here, and there's very few of the yellow, which are the CD8 cells here, and they're not appearing to get into the tumor. But here you can see the week 8 biopsy after this combination immunotherapy. Whether or not what component led to this and what these cells are recognizing is still an ongoing part of what my lab is working on. But as part of my conflict of interest management, I've updated this to be companies in the last 2 years are active. So collaborations that I have, I will talk about a clinical trial that Insight is supporting and which has the vaccine that UBVac has made. So what I think of when I think of what's happened in the last 3 years, these ideas, I'm just going to try to get rid of these little things, excuse me, that are, let me go back here. Apologies for 1 second. I'll try and close this up so I can see my whole screen and not have it be blocked. I hope I don't lose you guys. No, I'm not leaving. I just want to get rid of this. I want to get rid of that. I don't want to get rid of this. Okay, sorry.
Perfect.
So I think this dark matter is really the biggest discovery of the last 3 years, in my opinion. And this is, this is the cancer, and this is class 1 and class 2 molecule. I got to go back to my pointer, sorry. Okay, class 1 and class 2 molecules, and these are the peptides that are being presented. And what dark matter really is these peptides that we didn't know about on the surface of cancer. And so it's a rapidly evolving area. Many of these are derived from the other 98% of the genome, right? We're made up of that 1.5 or 2% so of our DNA, but these are other proteins that we didn't think were being expressed. Many of these are expressed only by cancer, not in the thymus. So these have been considered not mutated, alternative cancer neoantigens. This is a term that Kathy Wu has used. And while I think what we're focused on here are the ones that are expressed by cancer and not in the thymus, it seems clear that these non-canonical epitopes are associated with many other diseases. So there's different epitopes potentially that are associated with autoimmunity and other things. Some appear to be shared by cancers. That, I think, is what is really exciting. And they appear to be epigenetically regulated. And some of these are associated with bad outcomes. So some of these may turn out to be like driver mutations that in cancer. So why are we just discovering these now? Well, it's a relatively new area of science, and I'll go into that. It appears that they're not recognized by endogenous immunity. So if you vaccinate with whole tumor cells or TIL, look at TIL, they don't see them. So you need to prime in vitro or potentially vaccinate to generate these responses. So there's potential for novel T cell receptor and next-gen cancer vaccine. So I'm going to try and cover a big chunk of this here. So why are we just discovering them now? It's because we used an old-fashioned way of looking for things. We only looked for things that we thought were there. So you would go ahead and immunoprecipitate class 1 molecules, elute off the peptides, run them through a mass spec, and then you'd use something like UniProt or SwissProt, which is a database of the expressed genes. So when you do mass spec, what you get is a spectra of the amino acids that are there. And you've got to then use a database which has got the sequences that you can look at to figure out what those peptides are. So we only had a database that only had 1.5 to 2% of our possible genes. And that's why we were only seeing these canonical or these expressed genes. So the new approach is to have a database of the whole transcriptome, and there's different ways to assess that. But it's more complicated to develop this database of all translated proteins, and many of these are also out of frame. So you got to do it lots of different ways. So I was kind of shocked as I was reading into this more. This paper is more than a decade old, but it says that up to 80% of the human genome has the capacity to be transcribed into non-coding RNA. And the data indicate that there's additional protein coding genes that remain to be found. And I think that's been obvious. This is the other part of it, that this field of using mass spec to analyze the peptides, there were very few immunologists and very few people that were in this field in the first place. And so it's only been more recently, in the last decade or so, that you've had immunologists really digging down and having more of them looking at the kind of peptides that were there. I mean, there have been notable groups of people that have been doing it much longer, but there hasn't been that much. And it's only been this foundation of the Human Immunopeptidome Project, which only started in 2017. So this review I took and modified it, kind of ended in 2020. But I think these last 3-4 years have just been, amazing as people have studied the immunopeptidome that's on different cancer cell lines or freshly isolated cancer cells from patients. So to give you an idea, this is a recent review on micropeptides, which is what these are. And these are, many of these, actually all of these, are things like long non-coding RNAs or link RNAs or other components, which actually we didn't think were actually being translated, but are being translated and are being translated in cancer. So, and this just goes through and shows you some of the cancers that these are in. And I'm just going through this just to give you some reference to what these components might be. Here's some other examples and where they're located. And I think there's lots of questions like, are these non-canonical micropeptides that are in the mitochondria or in the nucleus, are they actually getting to the surface or not? And can they be crossed or presented on the surface of cancer? But that's another whole area. There's examples. This presentation, which is available on the SITC website in its free open access publication or presentation by Claude Perot, really just changed my whole perspective. It was a brilliant talk, and it's now 2 years old, but it's relevant to this. And I just to summarize it, what from this graphical abstract, is he was looking at AML blasts. And the beauty of that was that he didn't have to culture them. So you don't have the culture artifact. So he could take AML blasts fresh out of the blood of these AML patients. He could then immunoprecipitate the class 1 molecules, elute off the peptides, and run LC-MS mass spec. He then had a whole transcriptomic database of the AML transcripts. So he went deep. It wasn't just exon capture RNA-seq. This was full transcriptomic work. So then he could pull out all these peptides. Then he checked for those peptides for their expression in healthy tissues. And he found peptides that were either not expressed or low expression in these tissues. And he identified those as being tumor-specific antigens. He found that of those tumor-specific antigens, 85% were derived from abnormal transcripts. There was high intrapatient sharing, and they could induce CD8 T cell responses. And I may get this a little bit wrong, but he came up with 58 peptides across the HLAs that he looked at that essentially would cover 95% of all the patients with AML. So again, this website, the SITC Cancer website, it was part of the Surface Home program, Claude Perot's talk. You can get access to that. So then fast forward a little bit more, and this Nature Biotechnology paper came out from Kathy Wu and Aviv Regev on unannotated proteins expand the MHC-restricted immunopeptidome. So this is unannotated proteins are non-canonical peptides are dark matter. That's the kind of in general way we're looking at it. And they did the same thing. They did, they did RiboSeq here to try and look at what was being translated. So they had this new ORF database, this novel unannotated ORF database. And then they did the immunoprecipitation, did mass spec. And then they looked at different cancer types and they found what were the contributions to the peptidome. And they looked at, it's not. And then they looked at when they looked at the spectra, and that's what this was here, about 3.3% of the peptides were these novel unannotated ORFs. But some proteins, right, are really big, right? And so they're going to be, they're going to have more peptides that are going to be identifying a single protein. When you look at here, these long non-coding RNAs and these, the ORFs, these out of frame peptides, they represented, well, they were only, they represented 16% of all the detected peptides on the surface of the cancers. So, so while they only were 3.3% of the peptides, when you looked at the unique proteins, it was 16% of them. So that 16% number, if you look at data we presented then later that year, we came up with 16% in our early preliminary data, and it wasn't as deep as theirs, and we're now going much deeper into the peptidome and head and neck cancer. But that 16% number was this from this full cryptic database of dark matter and non-canonical peptides. So that was November of 2022. In January, it was at the Center of the Cancer Scientific Summit. And I heard this guy, William Fried Pastor, who was a new faculty member at Dana-Farber. He'd been a postdoc at the Broad MIT, and he was looking at non-canonical peptides in pancreatic cancer. And he was using organoid cultures of pancreatic cancer, and he came up with 5 peptides that were shared by a number of these pancreatic organoids. He went on, and I'm using this with permission. He allowed me to tweet it and all. And this data has not been published as yet. But he then took these non-canonical peptides and he stimulated peripheral blood. And this one, NuORF number 7, which is an HLA-A201 restricted peptide, he generated a T cell receptor that was essentially T cells were 78% against that peptide. They isolated the T cell receptor, and actually, as part of a collaboration that's funded by Stand Up to Cancer between his lab and Phil Greenberg already had that receptor a year ago. And so as of April at AACR, he told me he hadn't started anything with that. But this is how I think, here you got peptide and pancreatic cancer shared by 5 of these pancreatic things. Now you've got the next couple of days, you had Kathy Wu talking at Keystone on this endogenous antigen presentation and T cell receptors against them. So I think there was a lot going on. Then this paper came out, and this paper was somebody else who trained at the NCI. Elaine Gross was also in Steve Rosenberg's lab, and she was looking at this non-canonical or dark matter or HLA tumor ligands, she was calling it. But again, it's all that. What she did is she isolated and identified these non-canonical peptides. She then took those peptides and she saw that the TIL from a patient didn't recognize them, so they weren't being seen. But if she took a peripheral blood from that patient and stimulated them with the peptide, she could get peptide reactive T cells against the non-canonical or dark matter. Then she looked to see whether or not they could see the patient's autologous tumor. They could. And then she went, and these are against this 5' HOX or this 5' UTR, ZKS, I don't remember what the genes are. But what she showed is that this one T cell receptor could see ovarian cancer, melanomas, and in this case, a GI tumor. And here's another different T cell receptor it could see across those 3. So you got one T cell receptor against a non-canonical peptide antigen can recognize melanoma, ovary, and gastric cancer. That kind of was amazing. So why weren't these non-canonical antigens found earlier? I think it comes down to work that most of these non-canonical epitopes appear to be what are called short-lived proteins. And maybe they also, Jonathan Udell might call some of them DRIPs or defective ribosomal products. But these short-lived peptides or proteins, they get expressed, they have some effect, and then they get degraded. So they're not around long. Long-lived proteins I've got identified as these red squiggly lines. They're more structural, or they're around longer, so that when a tumor cell dies, these are the proteins that get released for the APCs. And there may be 1 or 2 of these things laying around when an APC tries to pick it up. But that's not what's driving the response. So you get T cell responses against the red antigen, which are the red T cells, but the yellow T cells aren't seeing any peptide, the T cells that could recognize the yellow, because they're not getting cross-presented by the APC. So the short-lived nature of the non-canonical peptides doesn't allow them to prime immune responses. And this is probably why we hadn't found them either. And tumor infiltrating lymphocytes don't recognize them, which is why we didn't see them when we did all those gene screens, where we took whole cDNA libraries and put them into to targets and tried to drive them. And the same thing for mice that were immunized with whole tumor cells. You didn't prime immune. This is the hypothesis. You didn't prime immunity in those cases. So again, what Elena Gross did was they took first the tumor cells, they lysed them, they immunoprecipitated the HLA, eluted off the peptides, did LC-MS mass spec. They used a different database system, which is called Peptide Prism, which a group out of Würzburg developed, which is really a brilliant thing as well. And then they used that to filter for the canonical and non-canonical peptides. And they found that the non-canonical peptides, they then took those non-canonical, they called them non-CTL, but they could then take this yellow peptide that they pulled off of here, they could mix it with the APCs, they could mix the peripheral blood together, and now they expanded these T cells that could recognize the yellow cells. And those are the ones then that went back and, some of them, the two that they published, could recognize across those three histologies of cancer. So for me, it really turned the world upside down. I mean, this is kind of remarkable, especially in a world the last couple of years where the focus has been like strongly on neoantigens. And you think of it, I think of it as kind of the pendulum. The pendulum had swung way to neoantigens and was kind of swinging away from all these shared or other types of epitopes. And this is kind of fitting, kind of bridging the two together, because now you've got something that's shared, not expressed in the thymus. So like a neoantigen that's a newly mutated epitope, it's an alternative cancer neoantigen. And so I think for me, it's really turned the world upside down. And it's why I think it's the biggest discovery in IO of the last 3 or so years. So it led to this review or preview by Sierra and Wu, which talked about these non-canonical peptides, a new class of neoantigens, and the potential for universal off-the-shelf immunotherapies. And in this review, they're not talking about all cancer. They're talking specifically about the Claude Perot paper and about AML, but having a universal vaccine for AML. But in reality, based on the work that I just showed you from Lena Gross and all, I think the possibility exists that there are a limited number of pathways for tumors to be rapidly proliferative or undergo metastatic potential. And in fact, there are papers coming out that are showing this, right? That there's common pathways in breast cancer and other cancers that appear to be controlled by some of these long non-coding RNAs or 5' UTR type proteins that have an effect and then are degraded, but their effect goes on and is associated with malignant potential. That's an evolving area. So it's not just neoantigens. We've known this for more than 20 years, right? This paper that came out of Steve's lab, Yutaka Kawakami, majority of HLA-A2 TIL recognized shared melanoma antigens, and that if you recognize GP100, if the HLA-A2 TIL recognized GP100, it correlated with clinical response. Now, it doesn't mean that those are the cells that were mediating response, but unless you're looking at all these things, you're not going to figure that out. Some work by Mark Davis and Christopher Garcia, looking really hard at two men with HLA-A2 colon cancer. They characterized only 4 T cell receptors, and 3 of those 4 T cell receptors recognized a non-mutated peptide that's overexpressed in colon and other cancers. And then this paper out of MD Anderson, looking at the CG-like-1 antigen, which is shared by ovarian, breast, bladder, gastric, lung, and other cell lines. I just show this as that just to come back and bring up that this isn't a new thing. We've known there have been other shared antigens, but we've kind of, I think in many cases, not talked about them in classes to medical students or even to PhD students. And I think that's been a shame, actually. And then, so it's not just me thinking this. I think this was at the Parker retreat, September 13th of this last year. And it had this panel with Kathy Wu, Nina Bardwaj, Larry Fong, Patrick Ott. And I think in general, it was clear from this group. I mean, Steve Schoenberger was primarily focused on neoantigens, but the rest of them were all into this discussion and Han Chong To from Singapore. We're talking about shared antigens, that it's more than just neoantigens, and we've got to be paying attention to that. I'm not against neoantigens. I'm working with Eric trying a very small role in this work with neoantigens. But if you look at tumors, in this paper from Chalmers et al. In Genome Medicine, looked at 62,000 cancer specimens, there are very few that are really TMB high. And if our goal is to develop immunotherapies that are going to work for everybody, then we've got to be thinking about things besides just neoantigens. So, 1023. So it's more than just a little summary at this point. So it's more than just mutated neoantigens. Cancer's immunopeptidome, canonical and non-canonical, is a rapidly evolving area. Likely some of what I just told you will not be true in 6 months or a year, but it's what's rapidly evolving. Non-canonical are particularly relevant for cancer immunotherapy, I think. Some are expressed only by cancer, not in the thymus. That's why they've been called non-mutated alternative cancer neoantigens. Some appear to be shared by cancers. Some appear to promote proliferation, metastasis, and invasiveness. Why are we just discovering these now? The LC-MS field is expanding with an increased number of investigators. Some of the non-canonical peptides are not recognized by endogenous immunity. This, I think, is one of the biggest questions for our field, one that we're trying to address in head and neck and lung cancer. But it's one of those questions. I don't know how that's going to evolve. It appears that you need to prime in vitro or vaccinate to generate immunity against many of these non-canonical epitopes, but they are immunogenic. Early cancer antigen discoverer used TIL or T cells from whole tumor cell vaccinated animals or to look at antigens. And it appears, based on the work again of Elena Gross and some others, that there's probably not much expansion of these T cells that are reactive to non-canonical epitopes in a patient with cancer or a naive patient. Evidence that T cell responses to these non-canonical peptides can recognize multiple cancers is a game changer for me. I mean, this is, I think, really important because there's more than just these antigens that, I mean, the discussion over the last couple of years, at least I'm thinking back to things when Cell Genesis was doing their GVAC studies, and there were large numbers of people on the ed board, and we would always be talking about adenocarcinoma of the prostate, of the breast, of the stomach, of the lung. They're all adenocarcinomas. They all have these same overexpressed genes. These are targets. These are things that could be used, and I think it's coming back a little bit to that. Again, data is limited. And so what I've just said is going to evolve, and non-canonical peptides may not be presented by all cancers. We don't know, but I think there's clear potential for immunologic, and there's clearly potential for pharmacologic interventions, because as we think about these long non-coding RNAs, circular RNAs, other things, those things could be targeted with pharmacologic intervention. So there. So I'm now going to switch gears a little bit. I gave you a lot of background. I'm going to now focus a little bit more on some of our work. So our own efforts over the past 2 decades have been influenced by the work of Jonathan Adell. And this review that he wrote almost 2 years ago, is really a superb review, and it gets at many of the questions that we're trying to address in cultured cells, but also in freshly excised tumor samples and in patients that have had radiation, and patients and how they're controlled epigenetically, either with agents that might influence that in terms of treatment. And what we had done based on some of the work of Jonathan Adell was, this is supposed to be a cancer cell, and this is supposed to be a class 1 molecule, and the little circles are supposed to be the peptides. So what we want, right? We want a T cell response against these peptides. But in our vaccines, if we vaccinate with whole tumor cells, and that's what we had done, we've been studying GVACs for many years, and we're involved in a number of clinical trials with GVACs. These peptides, unless they were long-lived, they weren't going to get picked up by dendritic cells unless they had cross-dressing, where a dendritic cell plucks out the class 1 molecule with peptide and puts it on its surface. What it really needs is, it needs these proteins, these slips and drips, these short-lived proteins, defective ribosomal products, which get degraded rapidly through the proteasome, shunted through TAP in the air, and go later to class 1. So the idea that actually Hong-Ming Hu, a colleague of mine, came up with was to block the proteasome, shunt the slips and drips to the autophagy pathway, and then prevent the autophagal lysosomal degradation of those. And these peptides, the proteins were then contained in this autophagosome, and it contains a spectrum of these proteins. And it also, we're finding, also concentrated the non-canonical proteins. We didn't know that at the time, but our more recent research has demonstrated that. So going back a long time ago, they showed, we used GVAX models, which were GM-CSF secreting tumor vaccines that had been irradiated. This was looking at 3LL GM-CSF, which was our gold standard, pretty much. And when you use the autophagosome vaccine here, it was with dribbles, it was much better in 3LL. And it raised the question, sorry. So I wasn't sure about about this, right? But as we were thinking about it, the reason I spent about 25 years of my career working in autologous tumor vaccines and autologous tumor therapies, like TIL or tumor vaccines draining lymph nodes that we would expand with, was because of this work of Brennan Main, where you're likely familiar with this. But if you take 10 identical mice and inject them all with 3-methylclanthurine, which is a chemical carcinogen similar to cigarette smoke, and you put that in with peanut oil intramuscularly, they'll develop sarcomas if you use the right amount of everything. And if you take those sarcomas, you look at them histologically, they all look identical. If you do gene expression profiling, you see the gene expression profiles are really similar, but they've got unique neoantigens. And Bob Schreiber has studied this a lot and used it to look at his system. But if you vaccinate with tumor A, one of those tumors, vaccinate 100 mice with irradiated tumor A and come back 2 weeks later and challenge with a minimal tumor dose, TD100 for tumor A, they're protected. That's what the black box is. But you challenge them with any other sarcomas, and essentially they're not protected. And it's the same all the way down the line. So we asked the question, if you had a vaccine that concentrated these shared antigens, which is what we thought we were doing, and you did this kind of a system, you should be able to change this, right? So what we did is we created our own methylclanthurine-induced tumor system. We called it the MCA300 series. And we either injected mice with irradiated 304 cells or autophagosomes from 304 or 309, and we did it with 4 or 5 different tumor cell lines. We would wait 14 days and come back and challenge with a minimal tumor dose and then measure tumor survival. And so when you do this with the whole tumor cell vaccine, we replicate the work of Brennan Main. This was PhD work of Chris Twitty. The X's are experiments he didn't do. So, vaccinate with 304, protect from 304, but not from these other tumors. 309, you protect from 309, but not the other tumors. When he used the autophagosome vaccine, it was different. And I'm going through this quickly because it's all been published more than a decade ago. But if you vaccinate with MC304 autophagosome vaccine, you have significant protection from 304 and 310 and 311, but not from 309. But in the other 8 of the 9 potential combinations, you've got significant, what we're calling cross protection. Now, it wasn't as good. This is 95 to 100%. This, I think, is 60 to 80. This 20 to 40, but it's all statistically significant. So now if you look at the mice, right, if the mice you vaccinated, I'm showing you this is now with MCA304, because MCA304 vaccinated mice, they were only protected from 304 if you use the radiated whole tumor. If you use the autophagosome, it protected these two and not 309. But if you look now at the immune response you generated in these mice, if you just give them whole tumor cells, you now see a specific response to MC304. This is data that has been reproduced like 100 times in other labs with other tumor systems. When I was in the surgery branch, Jim Yang, Jim Muley, different people were using the MCA 102, 105, 106 to do all the TIL studies. They were all uniquely and exquisitely specific. It goes along with Bob Schreiber's data. This is all perfect, right? You vaccinate with the tumor, you get a response against that specific sarcoma, not any other sarcoma. Now, if you did the autophagosome vaccine, very different picture, right? You vaccinate with autophagosome vaccine, you now get an immune response to 304, 309, 310, 311. Now, remember, you didn't get as good a protection as you got with the whole tumor vaccine, but we turned on T cell responses against the tumor. That kind of was very interesting. So that was an interesting experiment, but it's not so relevant clinically because that's protection experiments and we're not doing experiments. We're not trying to treat patients before they get the cancer. Someday we'll do that, but so we wanted to switch it, and it was also in a syngeneic system, right? And we're thinking, can we do it in an allogeneic genetic system off the shelf because these should be shared. So some other work that Chris Twitty did with Sean Jensen and was later published by you and colleagues, but we did it in 3 different breast cancer models. We took them in 3 different mouse strains. We did it in a brown mouse, black mouse, white mouse, so BALB/C, Black6, C3H. And we made vaccines from all those different breast cancer cell lines, and we found that any breast cancer cell line, even if it didn't share any H2 with the mouse, could provide protection or could provide therapeutic effects if you combine it with a T cell agonist. In this case, we were using a vaccination with an anti-OX86, which is anti-OX40, and you could see that while no treatment didn't do anything, vaccine alone could do a little bit, OX86 could do a little bit, but the combination could have a big impact. And the allogeneic vaccines with OX86 were very strong. So that was one step. Another thing we found out, which we actually went to try to do combinations in models which were not PD-1 responsive. In our hands, we find lots of the more poorly immunogenic mouse models were not affected by anti-PD-1. But there were some that were affected by OX40. So OX40, a T cell agonist, right, that drives, could have an effect. And one of those models was this MMTV, PYMT spontaneous mouse tumor model, where we were transplanting the spontaneous tumors into other FEB mice. And then we were giving them OX40, anti-PD-1, or the combination. And what we found out is that, again, OX40 or PD-1 alone or no treatment had no effect on tumor growth. OX40 had a significant impact, but when you added PD-1, it made it worse. And you're just seeing this one thing here, but we did it with 3 different anti-PD-1 antibodies, 2 different PD-L1 antibodies. We repeated the experiment multiple times. It was part of David Messenheimer's PhD thesis, and that was a real effect. So we started playing around with what happened. I'm not going to go into it because it's published, but I want to share with you this point, that for reasons that we discussed in the paper, we delayed the PD-1 administration. And what we found out was that we had this way big, significant impact on tumor growth. And what we saw was that we were actually curing some mice in a model system. And Lisa Cousins was on David's thesis committee because she's a faculty member at OHSU, and she'd never seen mice get cured like this. So we shared all this data with all the pharma companies developing these reagents under CDA before any of this was published, and just to share that. So based on those data, we got very excited about the potential of vaccines and combination immunotherapy to have an impact. With NCI funding, we created a clinical product. It was an SBIR phase 1/2. We picked, in a collaboration with the FDA, working with Raj Puri, who at that point now more than a decade ago had the largest gene expression profiling arrays. We screened, I think, 20 cell lines I developed in my lab to find ones that had the greatest separation of Venn diagrams that would cover the greatest number of different potential cancer antigens. And we looked, and we ended up with 2 different cell lines, which were an adeno and then a squamous mixed histology. And that covered the spectrum. The product we made from that comes out, it's these autophagosomes that are double membrane microvesicles. On their surface are spectrin and F-actin. Spectrin F-actin are ligands for CLEC9A, which is on the cross-presenting DC. This is an EM, looks like. The tumor cell lines, we got really lucky here. The tumor cell lines we picked also had natural TLR nod agonist activities. They had 15 DAMPs, things like heat check proteins and calreticulin. They've got more than 100 putative cancer antigens for a spectrum of cancers, and it includes these non-canonical or dark matter, and that we've characterized more recently by mass spec. We had an adjuvant phase 1/2 that the NCI funded, which gave us formulation, proof of concept, and trial objectives for meta and long-term drug stability. So the concept is we give this vaccine, and we give it the first 2 vaccines intranodally under ultrasound guidance to really get more bang for the buck based on other preclinical data that I'm not talking about, but it buys us 10 to 100-fold better priming of immunity from that data. So this was the phase 1/2 that we did with the NCI. We did it in patients that had been definitively treated. I didn't think you should put any vaccine alone into a patient with advanced cancer anymore at this point with the failures that we had. And so we were looking to do it in an adjuvant setting where we could actually look at whether we induced immunity, which was our main goal. So on average, we've got about 300 proteins for patients with lung adenocarcinoma. This is number of patient samples and the number of genes that are overexpressed for proteins that we've got in the vaccine by mass spec. And so this is TCGA data sets. So we looked at them by B cell responses. We used initially in Vitrogen protein arrays, but have also used microaffinity proteomic arrays that Fridtjof Lund Johansen makes in Oslo, Norway. And more recently, the CDI FIP arrays, which have 270,000 phages with overlapping 90mers of the 29,000 proteins in the human expressed genome and some of the modified versions. We also evaluated T cell receptor responses and saw that we actually increased by 10x the number of T cell receptors in patients that were vaccinated. So why were we looking at antibody responses? Well, my lab's interested in this idea of coordinated B and T cell responses to cancer because, you can only, with a tube of blood, you can only do so many T cell functional assays. But with 1 microliter of serum, you can run protein arrays against the whole human proteome and every other antigen you could think of. Um, so the first paper, this Rena Queck working with Larry Fong, this GI paper more than a decade ago, was the first to show that if you had an antibody response, in this case, in men with prostate cancer who were getting CTLA-4 and GM-CSF, that they had antibody response against PAX6, and that correlated to T cell response against it. This paper by Tripathi and PNAS in 2016 is an amazing paper. It's got a bad title, but it's amazing data, and it shows that patients with lung cancer that are newly diagnosed, no treatment, have got antibody responses to a whole host of the overexpressed genes that are where peptides are present on the surface of the cancer, and they've got T cell responses against it if you look at their peripheral blood. And then I'm going to talk to you briefly about another PhD student in my lab who did this in the mouse model. So essentially, we wanted to demonstrate what we've been trying to do in the human, and it was very hard, so we did it in the mouse, that the response of T and B cells was coordinated. A little quicker, but essentially he vaccinated mice a couple times, and then he looked for serum and T cell responses in mice after a couple vaccines. So what you're doing here now is we took 4T1, we sequenced 4T1, and we had what we thought were the neoantigens. We then looked at the publications on 4T1, and where we found the same mutation that others had found, we had peptide arrays made, 15 mers, that centered the mutation in the 15 mer. And then we also made the wild type 15 mer as well. So we had, I think, 90 the mutations covered with either the mutant and the wild type. Now you use that to identify in the individual mice what antibody, what antibody, what's the antibody targeted, which of the 15 mers it saw, and then we went back in the same mice and we looked at the same mice what peptide they saw. With there we use not the 15 mer, we use the minimal peptide, the H11 mer. And what you see is that there's a nice slope here, and you've got a strong antibody response against DPP9, which is the wild type epitope, and here's the mutant epitope, DPP9. So there's a little stronger antibody response and a slightly stronger T cell response. Other ones, you have the mutant epitope much higher than the single nucleotide variant, much higher than the wild type epitope for that. But so this was part of it. And then the gold standard is not just that you see the peptide, but that if the IgG signal intensity can also correlate with response to the naturally presented peptide by the tumor cell line here in case of 4T1. So that's why we're looking at antibody responses, and in fact, they correlate. So now let's go back to that lung cancer trial, which was the adjuvant study in patients that had been definitively treated with lung cancer. So targets where 2 or more patients made antibody responses to the targets that are associated with poor outcomes. So here, these proteins are all genes that are overexpressed, can be overexpressed in lung cancer. When they're overexpressed in lung cancer, the survival is the red line. The blue line or the purplish line is the total TCGA subset for lung adenocarcinoma. So if you had an overexpression of CDK5R1, you did worse. So we had 2 or more patients that made, of the 13 patients we vaccinated, that made antibody responses to CDK5R1, to CDC42-BPP, to ACP1. So we're in the process. We're still in the process of trying to characterize whether there's T cell responses against those. But this is why we think it's relevant. One of the targets that we have characterized response to is NCOA4. Here we're looking at 2 different ways of assessing antibody responses. This is using a FIPR or a microaffinity proteomic array. This is looking at protein arrays. I'm sorry. This is the protein arrays here is 27.4-fold, and this is the amount of this protein that's present in the vaccine in terms of relative protein concentration. So we had an antibody response that went from this to 12-fold to 13-fold, and by 27-fold increased antibody response to NCOA4. And when we looked at the antibody response over time, and then we looked at T cell responses, there was a T cell response against NCOA4 that we've characterized. So it isn't just antibody responses to proteins that are overexpressed in lung cancer, but there's also antibody responses to these same proteins that are overexpressed in head and neck and in triple-negative breast cancer. And we did initiate 2 trials in both of these, but I'll focus on head and neck for a second. We had a patient with lung cancer that I just talked about, and we drove his cells and isolated the cells that were tumor reactive. So we picked out by 4-1BB these cells. And so you can see they're making almost 10 nanograms of interferon gamma when we mix them with his autologous tumor. FADU and SEC9 are 2 head and neck cancer cell lines from ATCC that share different HLA with the autologous tumor here, this patient's tumor. His T cells recognize both these 2 head and neck cancer cell lines that share HLA, but not this head and neck cell line that did not share HLA. So these data stimulated our interest in identifying the cancer immunopeptidome. And that led to this collaboration with Takashi Shimada and Noriko Uemoto, who are at Shimadzu in Kyoto, Japan, and Yuriko Minagishi, who's at the Japanese Foundation for Cancer Research in Tokyo. And then people at the Institute here, Ryan Mang is one of the data scientists at the Institute, and Brian Pining heads that group, and Tracy Hilton is at UBVac and has been doing the mass spec for UBVac. So kind of going a little bit further, so we have complex vaccines. So we're not the only group that's looking at complex vaccines. Liz Jaffe's group at Johns Hopkins with Wei Zhang have been looking at GVAX with T-cell agonists. So we were looking at OX40. We also looked at anti-GITR, but they were also giving anti-4-1BB and anti-PD-1. So they had data that supported that as a strategy. I mentioned these data, which were the data with anti-OX40. We also had data with anti-GITR. This led to clinical trials where we had a trial with BMS, which was vaccine plus OX40 and PD-1, delayed PD-1 in triple-negative breast. We had this trial that's going on in head and neck cancer, and then Liz Jaffe had the trial in pancreatic cancer. Unfortunately, BMS stopped developing agonists because agonists and PD-1 alone haven't had much of a clinical effect. But fortunately, Insight our timing was reasonable, and so we were able to pull off this trial, which we've completed the first part of this now, and we were giving either vaccine with delayed anti-PD-1 or vaccine with anti-GITR with delayed anti-PD-1. We were taking serial biopsies, pre-treatment, week 2, week 8, and the goal of this was to, of course, look for safety, but also to explore immune correlates and look at response rate and overall survival. So I want to talk to you a little bit about our monitoring and what we've been very excited about on this. And so the serial biopsies have allowed us to take 4 TrueCut cores on 17 of the 18 patients and do histology on 2 of them, and take the other 2 cores and disassociate those and isolate the tumor cells so we could do single cell RNA-seq and site-seq, T-cell receptor and B-cell receptor analysis, as well as try to develop autologous tumor cell lines and things there. At the same time, we're also looking in the peripheral blood. We're looking at doing single cell sequencing as well. Here, T-cell receptor, B-cell receptor analysis, but we're also doing FIP arrays, looking at antibody responses and flow. So just kind of give you a high-level overview of what we see, and this is from arm 1. So this is a patient that gets vaccine for 2 weeks and then starts on day 15 with anti-PD-1. And essentially, if you're looking here, we're focusing on CD4 effector memory and CD8 effector memory here. And on the upper right quadrants of the flow histograms, you're looking at activated cells. So these, in the case, are HLA-DR positive and CD38 positive. And you can see that even with vaccine alone, we're seeing increases in the activated effector memory cell populations that appear to continue with time. So another project that we had with my colleague Andy Weinberg, and I believe was driven by Thomas Doohan, was looking at CD39 and CD103 in TIL, and showing that those are recognized autologous tumor. That's data that we've done in collaboration with our group to see that if you isolate those cells, they do recognize autologous tumor. But the patients who have more of those cells are the ones that do better. This is looking at all head and neck, and this is looking at HPV negative head and neck cancer. So these CD39, 103 double positive T cells are associated with improved outcome And in the work that was published, we show that they are enriched for tumor-reactive T cells. So what we've seen in the patients that we've vaccinated so far is that we are seeing that if you look at the CD39 103 double positive cells, that they're increasing by week 8. And if you look then, so that's the first thing. The second thing is, as we've done the single cell sequencing and then gone back, and that'll give you the percentage of the cells. And we're to do the single cell sequencing, we're isolating the tumor cells, we're enzymatically digesting the tumor, then we're sorting for CD45, and then we're doing 10x on that. So what we do is we get the gene expression profiling on the CD45 positive cells. But what you want to do is you want to figure out what does that relate to back on the tumor section. So what we've done is this, what we're called this adjusted density or proportional adjusted density or PAD. And that is we do multiplex IHC on the tumor samples. We know how many CD3s there are per millimeter squared. We know in the 10x single cell sequencing data what percentage of the CD3 are granzyme B and or interferon gamma positive, and we can look and then extrapolate back to the tumor sample to see how many of the cells in the tumor biopsy are granzyme B and or interferon gamma positive. And you can see that they're going up substantially in these patients. Okay, so sorry. So we then looked at multiplex, and we've also seen that things like LAG3 go up. So if you look, I'll just go to this green line is CD8 LAG3 positive per millimeter squared. So you can see in this patient who got this side got vaccine, and then PD1, this got vaccine getter PD1. But you can see that LAG3 goes up, LAG3 goes up, LAG3 goes up. This one, it doesn't go up as much, but it's still going up. So LAG3 is going up. When we drill down on this a little bit, and you're really focused on, this is now looking at the 10x data, and if you focus over here on the biopsy, you can see that by week 8, the total T cells are up significantly for LAG3 expression. So this is looking at all T cell receptor positive cells in the 10x data. So you see LAG3 is going up. But if you drill down on that and look at the cells that are CD39 103 positive, those are the cells that should be the tumor reactive cells in our opinion, right? You can see that their expression is up significantly by week 2. And so if you look at the patients who only got vaccine by week 2, which are GDP13 and GDP01, you can see they're going up essentially some of the greatest degrees. So this effect here between pretreatment week 2 is an impact of 2 vaccines, which is given at day 1 and day 8. So that's what we think is driving some of this response. So based on those data, we actually have a study that on arm 3 that's being added to this trial, which is going to be vaccine plus anti-LAG3 plus anti-TIM3, which will then go in with delayed anti-PD1, and Ram Widener will be the PI, and that's being funded by NCI. So I want to come back in the last couple minutes to, because I know I've gone through a lot. I figured I could do that with a group from the NCI, but this is a patient who was a lung tumor patient. It was patient 02, and this is looking at mass spec of the proteins that are contained in the 2 components of the vaccine. So you see that there's about 800 proteins for which LT101 has a peptide on its surface, right? And if you look at what those peptides are, about 8% of the peptides on its surface are non-canonical. So we're trying to figure out, did this patient make a T cell response against the non-canonical peptides? And I don't have the answer for that to you today, but I want to show you where we started to drill down on this. We ran the FIP arrays, which again have, so this vaccinated patient, LT02, makes antibody responses to a large number of canonical proteins with a peptide that's presented by HLA or autologous cancer, right? So they got an antibody response. I showed you earlier that we could correlate antibody responses with T cell responses. We've done protein array, which is whole protein, versus FIP, which is 90 mer linear peptides. We have at least a 4x increase in antibody response to 136 proteins, which are really 121 unique proteins. By protein array, it's about 100. And then of those, there are 58 that we know there's a peptide on the patient's cell line, or 28 here. So we're trying to correlate that. So these antibody responses are to the canonical proteins. So we also have identified 753 non-canonical peptides that are on the surface of this patient's cancer cell line. The majority of these are 5' UTR, but there's a good number that are off frame. So, and again, this is with collaboration with the group from Shimadzu and from the Japanese Foundation for Cancer Research. So FIP has detected antibody responses to 60 canonical sequences where a 5' UTR epitope for that protein was identified and is being presented by the HLA of the patient's tumor cell line. So we're thinking of intramolecular epitope spreading, and that's part of where we're focusing the initial look. And we're focusing our T cell function studies on those 60 non-canonical peptides. So we started with manufacturing the peptides, and then we have another project that's using RNA mini genes to put those together. So my preliminary summary, I won't get into the last part. I'm just going to, in the first 4 patients evaluated, and we've now got a few more, but it's tumor infiltrating T cells at week 8 increased from pretreatment levels by an average of 4.3-fold. The density of these 39103 double positive cells previously shown to identify tumor reactive T cells also increased in all week 8 biopsies. The week 8 biopsies have increased numbers of cells expressing interferon gamma and granzyme B. They also have increased numbers of T cells expressing LAG3 and TIM3. I didn't show you the TIM3 data, but the TIM3 data and the LAG3 data were notable enough that NCI is giving us both those antibodies for the next arm in the clinical trial and funding the trial. Preliminary T cell receptor evaluation of the tumor identified proliferation of clones previously undetected in the peripheral blood, including alpha beta T cells, iNKT, and MATE cells, and we're currently evaluating those. An expansion of clones that predated treatment was also identified. So what I've talked to you about today has been work that's been by a lot of people. I've tried to identify them along the way, but people in my lab, Sean Jensen, Tarsen Mudville, Christopher Postian, Quinn Vu, have all been working on this. I've mentioned other past PhD students and fellows who have worked on it. It's been a strong collaboration with Hongming Hu, whose lab, Jana Patel, and others have worked on this project over the years. A lot of the work that is the monitoring data is run out of my colleague Yoshi Koguchi, an MD PhD, who runs a big chunk of our immunological monitoring group. Our pathology colleagues who are in charge of the molecular genomics and components and help us with bioinformatics and other projects. My clinical colleagues, most notably Walter Erba, who's the director of the Institute and has been supporting all this effort for the last almost 30 years. Ron Widener, who leads the current clinical trial, but others who have been components in other clinical trials that we've run, collaborations with our friends in Germany, with Oslo, I mentioned for Truffle Johansson, who did microaffinity arrays, Shimadzu and JFCR, who are doing the mass spec with us, and some others. And then, again, some of our funding sources, but most notably to the patients who believe in what we're doing and take tours of the lab and meet the PhD students and the fellows, and who are the bravest people, right, to go in and donate their blood and their biopsies. So I thank them and all the nurses and all, and I'll stop there. If I haven't put you all to sleep, I'd be happy to take questions.